Clarity Clinical Solutions.
Are children just small adults? When a new drug is developed, it's tested in adults first, sometimes for years, before anyone even thinks about giving it to a child. But here's the problem: children are not miniature adults. Their bodies are growing, their organs are developing, and their metabolism changes almost yearly. Giving a child the adult dose scaled down by weight isn't just imprecise, it can be dangerous. So how do we figure out what's safe? That's what pediatric clinical trials are for, and they come with their own unique set of challenges and safeguards. Let's start with the biology. Children's bodies process drugs completely differently than adults. The liver enzymes that break down medications develop gradually over the first years of life. A newborn's kidney function is only about 30% of an adult's. Their body composition, more water, less fat, changes how drugs are distributed. And then there's the brain. The blood-brain barrier isn't fully formed in infants, which means some drugs that are safe for adults can be toxic in babies. On top of all that, children are growing. A drug that affects bone growth, hormone development, or brain formation can have consequences that last a lifetime. So how did we handle this in the past? Honestly, not very well. For most of the 20th century, doctors simply guessed. They'd take the adult dose, divide by weight, and hope for the best. The results were often tragic. The most famous example is the sulfanilamide elixir disaster of 1937, where a toxic solvent killed over 100 children because nobody tested the liquid formulation. Then in the 1950s and 60s, thalidomide caused severe birth defects because it was never tested in pregnant women. These disasters led to modern drug regulation. But even then, pediatric testing remained rare. As late as the 1990s, over 70% of drugs prescribed to children had never been tested in children at all. That started to change in the late 1990s and early 2000s, thanks to two landmark laws in the United States. The first is the Best Pharmaceuticals for Children Act, or BPCA, passed in 2002. It gives drug companies an extra 6 months of market exclusivity, basically, patent protection, if they voluntarily conduct pediatric studies. The second is the Pediatric Research Equity Act, or PREA, passed in 2003. This one is mandatory. It requires companies developing a new drug to also study it in children, unless they get a waiver. Together, these laws have transformed pediatric drug development. Before BPCA and PREA, only about 20% of drugs had pediatric labeling. Today, it's over 50%. So what do these laws actually require? Under PREA, when a company submits a new drug application for adults, they must also submit a pediatric study plan. This plan has to outline how they will test the drug in children, what ages, what doses, what safety endpoints. The FDA reviews this plan and can require additional studies. There are also requirements for pediatric formulations. If the adult version is a large tablet, the company may need to develop a liquid or chewable version for children. And importantly, the FDA can grant waivers, for example, if the disease doesn't occur in children, or if the drug is likely to be unsafe. But the default is, if adults take it, children need to be studied too. Not every drug needs pediatric testing right away. The FDA has two important tools: waivers and deferrals. A full waiver means pediatric studies are completely excused. Maybe the condition doesn't exist in children, or the drug would be unsafe at any pediatric dose. A partial waiver might excuse testing in certain age groups, like newborns, while requiring it in older children. A deferral, on the other hand, means the studies are required, but they can be done after the adult approval. This is common when a disease is rare in children and it would take years to enroll enough patients, or when the adult formulation needs to be reformulated for pediatric use. Deferrals are important because they balance the need for timely adult access with eventual pediatric data. One of the most unique aspects of pediatric trials is consent. Children cannot legally consent to participate in research. That's a fundamental ethical protection. Instead, parents or legal guardians provide parental permission. But that's not the whole story. For children old enough to understand, usually around age 7 and up, researchers must also obtain the child's assent. Assent means the child agrees to participate in a way that's appropriate for their age and maturity. For a 7-year-old, this might be a simple conversation with a picture book. For a teenager, it's closer to adult-informed consent. And crucially, a child can withdraw their assent at any time. If they say they want to stop, they stop, even if the parents say it's okay. Parental consent comes with its own complexities. For most pediatric trials, consent from one parent is sufficient. But for higher-risk research, the FDA and IRBs may require consent from both parents, ensuring that neither parent objects to their child's participation. There are also special rules for children in foster care, where the state is the legal guardian, and for emancipated minors who can consent for themselves. And there's the question of therapeutic misconception. Parents may believe their child will personally benefit from the research, even when the study is designed to gather knowledge, not provide treatment. Researchers have an ethical duty to make this distinction crystal clear. Designing a clinical trial for children is fundamentally different from an adult trial. Sample sizes are often much smaller. There simply aren't as many children with a given condition. This means researchers need to use creative statistical methods that work with smaller numbers. Many pediatric trials use adaptive designs, where the dose or treatment can be adjusted based on interim results. There's also a greater reliance on extrapolation. If the disease is similar in adults and children, and the drug works the same way, regulators may accept data from adult trials combined with smaller pediatric pharmacokinetic studies. This reduces the number of children who need to be exposed to experimental treatments. Dosing is one of the trickiest parts of pediatric drug development. Children are not just smaller, they're growing and changing continuously. A dose that works for a 6-year-old may be completely wrong for a 6-month-old. Children go through rapid changes in liver function, kidney function, body composition, and metabolic rate. Many drugs need to be dosed by body surface area rather than by weight. Some drugs require liquid formulations, which need to taste acceptable, remain stable, and have appropriate preservatives. A drug that tastes bitter may be refused by a child, making compliance impossible. And then there are neonates, premature babies who weigh less than a kilogram, where a single drop of fluid can be a significant dose. Minimizing pain and discomfort is a top priority in pediatric trials. Children are not capable of tolerating multiple blood draws, long clinic visits, or uncomfortable procedures the way adults might. Researchers use a variety of techniques to reduce burden: topical numbing creams before injections, smaller blood volumes, child life specialists who use play and distraction, and the option to conduct visits at home rather than in a clinic. Some pediatric studies use pharmacokinetic scavenging, collecting small leftover blood samples from routine clinical tests rather than drawing extra blood specifically for the study. Every effort is made to make the experience as non-traumatic as possible, because a child's negative experience with research can affect their willingness to participate in medical care for the rest of their lives. Some pediatric populations are especially vulnerable. Neonates, babies less than 28 days old, have the most immature organ systems and the highest ethical protections. Research in this group is tightly regulated because neonates can't voice discomfort or assent. Then there are children with rare diseases, often excluded from large-scale trials because there simply aren't enough patients. The FDA's Orphan Drug Program provides incentives for studying these populations. And children with cognitive impairments require even more careful assessment of assent capacity. The rule is always the same: the research must offer a favorable risk-benefit ratio, with the lowest possible burden and the highest possible safeguards. Despite the challenges, pediatric trials have produced remarkable successes. Take childhood leukemia. In the 1960s, survival was less than 10%. Today, thanks to decades of pediatric clinical trials, it's over 90% for some forms. Antiretroviral drugs for HIV-infected children extended life from months to decades. The development of surfactant therapy for premature babies, tested in tiny infants weighing less than a kilogram, reduced newborn respiratory distress deaths by 50%. And the pediatric trial network set up under BPCA has led to improved dosing for everything from antibiotics to asthma medications. These are real, measurable impacts on children's lives, and they would not have been possible without pediatric clinical research. So let's bring it all together. Children are not small adults, their developing bodies, their legal status, and their unique vulnerabilities require a completely different approach to clinical research. Pediatric trials are governed by strict regulations, the Best Pharmaceuticals for Children Act and the Pediatric Research Equity Act. They require both parental consent and age-appropriate child assent. The trials themselves use smaller sample sizes, adaptive designs, and special techniques to minimize pain and discomfort. The result? Safer, more effective medications for the youngest patients, and a system that continues to evolve and improve.